In this video, we will study about regulation of gene expression. Now, what is gene regulation? Now, genes, they are part of DNA. They are also unit of inheritance. Genes also control structural component of an organism. Genes are also enzymes, but some of the genes, they are regulating the other genes. So this is called as gene regulation. So we can say, what is gene regulation? It is a mechanism of regulating or controlling the activity of other genes. Some genes will function and some genes will be repressed. So when they function, this is called as switching on, and when they are repressed, they are called as switched off. Gene regulation is of two types, positive and negative. Now, regulation of the genes can occur at the formation of the RNA, called as transcription, or after the formation of the RNA, modification occurs. This is called as post-transcription. Like if RNA has been produced and this has to be converted into messenger RNA, on one side cap will be added, on the second side tail will be added. So this is called as post-transcriptional modification. At the formation of the protein, translation. After the formation of the protein, post translation, like if insulin has to be produced, first there is proinsulin, then that will be converted into insulin. So this is called as post translational regulation. Formation of hormone. Then at the genetic level, genomic regulation. So genes, they are part of the DNA. They may be forming structural component, they may be forming enzymatic component, and some of the genes which are regulatory in function. For example, if we have a cell and this cell is undergoing cell division, so we are getting cells. Supposing we have got 200 to 250 cells. Now cell division has to be stopped, and then that is done by gene regulation. So genes which are helping in cell division, now they has to be stopped. So this is called as gene regulation, or this is called as regulatory genes. Similarly, if we take another example, like this is of cell division, we can take other processes like cell development, growth, differentiation. Supposing a cell is getting converted into xylem tissue or phloem tissue, after a certain time, this has to be stopped, and this is done by gene regulation. Like if we talk about digestion process, when we eat the food, digestion will start, digestive enzymes will be produced, and after digestion has been completed, this has to be switched off. So this is called as regulation of the genes, and that is done with the help of regulatory genes, which are a part of DNA. So on DNA, there is present a unit called as operon. So operon is a unit of the DNA which will control development, structure, and functioning of other organisms through selective transcription activity, that is by the formation of the RNA. Now this was studied by Jacob and Monod in 1961. They are French scientists, and they were studying the metabolism of Escherichia coli bacteria. Now they were studying lactose metabolism of Escherichia coli, and they found during that study that genetic material or DNA of Escherichia coli possess gene units called as operon. Now what is an operon? You can see here, this is the DNA unit. Now this is having regulator gene, promoter gene, operator gene, and these are the structural genes. So what is an operon? This is a part of genetic material which act as single regulated unit having one or more structural genes, operator gene, promoter gene, regulator gene, repressor, inducer, and co-repressor. Now what are these structural, operator, co-repressor, repressor? This we'll discuss in detail in the next slides. So operons, they are of two types, inducible operon system and repressible operon system. When the gene is switched on in presence of inducer, this is called as inducible operon system. And when the gene is switched off, this is in the presence of repressor, this is called as repressible operon system. So we can see here, this is the regulator gene, this is promoter, this is operator. These are structural genes. O is for operator gene, P is for promoter gene. Inducible operon, this require an inducer which will start the metabolism. Repressible operon, which will be inhibited in presence of repressor. So this is called as repressible operon system. So first we'll discuss about inducible operon system. Now, Jacob and Monod, they were studying metabolism of Escherichia coli in presence of lactose. Like if bacteria are growing and during its culture, lactose is added into the culture medium. Now, how this lactose is being utilized by the bacteria. So this they studied. So here we can see this is again regulator gene, promoter, operator, and these are the structural gene, Z, Y, and A. These are structural genes. So what is an operon? Operon is a combination of these genes, regulator, promoter, operator, and structural genes. And here lactose is acting as an inducer. In presence of lactose, this gene is switched on. But if lactose is not there, this gene will be switched off. So this is called as inducible operon system. Like what happens when lactose is added into the media, this regulator gene which is forming a inhibitor formation of RNA and then protein called as repressor. If this repressor is binding to this region that is called as operator region, then this RNA polymerase will not move and these proteins will not be produced. So when this lactose is there, this will induce this inhibitor from here and then RNA will be forming and protein will be forming. So this is called as inducible operon system. I mean, this is being induced in presence of lactose. So here we can see this is the regulator gene which is regulating these genes. Now this is producing the messenger RNA by transcription and that is forming a protein called as repressor protein. Thus this repressor protein will go and bind to the operator gene. Now this operator gene will not function when this has been blocked with the repressor. So this is called as repressor operator complex. And this is the RNA polymerase which is recognizing the promoter region. Now when this is blocked, here no function will take place. So this is called as gene is switched off. Now in presence of lactose, lactose is acting as an inducer. Now this will bind with the repressor. So when this bind with the repressor, this part will be free, which was blocked in presence of repressor. Now this RNA polymerase will move over the structural genes. RNA will be produced by transcription and these RNAs, they will produce the proteins. So these proteins will help in utilization of the lactose by the Escherichia coli bacteria. And when this gene is working, this is called as gene is switched on. Now we'll discuss one by one structural genes, promoter, operator, regulator genes. Now first we'll discuss about structural genes. As we have seen in the earlier slides, the structural genes, they undergo transcription and form RNA. Then those RNA, they form the proteins. There can be more than one structural gene. Like in Escherichia coli, we have seen there are 3 structural genes, Z, Y, and A. And we can see here, Lac Z, Lac Y, Lac A. Now when this operator is free, this is not blocked by repressor. Now this Lac Z will be undergoing transcription. RNA polymerase will move here and this will help in transcription. Now this RNA is polycistronic, so we'll get Lac Z. This is transcription from Lac Z. This is from Lac Y. This is from Lac A. So these RNAs then with the help of ribosomal RNA and transfer RNA, they will form protein. 3 kinds of proteins will be produced. One is beta-galactosidase, second is lactose permease, and third is trans-acetylase. These are enzymes. I will study their role in the next slide. Lactose is converted to glucose and galactose in presence of beta-galactosidase. So these structural genes, they help in formation of these 3 proteins. So this is the function of the structural genes which undergo transcription, translation, and they form the protein like we discussed. So what is the function of these 3 protein? If we see lactose when added into the media, in the bacterial medium, now this will be converted simply into glucose and galactose by beta-galactosidase. Now if we have this Escherichia coli bacterium and we have lactose also in the outer medium, right? Now this has to enter into the bacterium. Now this will be done with the help of galactoside permease. I mean, this will allow the entry of lactose into the bacteria. So this is called as galactoside permease. And what is the function of galactoside acetylase? This help in conversion of beta galactoside to get acetyl group, and acetyl group is obtained from the acetyl coenzyme A. So transfer of acetyl coenzyme A, acetyl group from acetyl coenzyme A to beta galactoside. This is by galactoside acetylase. This is also called as transacetylase. So lactose will be converted to simple sugar called as glucose and galactose. Supposing this is the Escherichia coli bacteria, lactose has entered in here. Now this lactose will be converted into glucose and galactose. Now the simplified food will be taken by the bacterial group. So this is about inducible operon system in case of Escherichia coli. Now next we'll discuss what is actually operator gene, what is its function of the operator gene. Now this is the operator gene. Now only this operator gene is responsible for the transcription process. If RNA polymerase is recognized and this will move to the structural gene, then only gene is switched on. So we can say this is a gene which control the synthesis of messenger RNA over the structural gene. If this is blocked, this will not work. And if this is free, this only will work and this help in the transcription. So this is about the operator gene. Now, next is about what is promoter gene. Now we can see this is the promoter gene which is present before the operator gene. Now this is the place where RNA polymerase will recognize this promoter gene, and when operator is free, this will move to the structural gene and transcription will take place. So we can say what is promoter gene? This acts as an initiation signal which function as a recognition center for RNA polymerase, provided the operator gene is switched on. If operator gene is not blocked, then only promoter gene will function. RNA polymerase will recognize this area. This will bind to the promoter gene. Then this will move to the operator gene, then to the structural gene, and then transcription will take place. Then only gene will become functional. Now next we'll discuss what is regulator gene. Now main role of this regulator gene is regulate all these genes. So this is called as regulator gene. This will produce a repressor. Now repressor, like this will undergo transcription. RNA will be produced and this will produce the protein which act as a repressor. And if this is blocked, this part is blocked, here it is RNA polymerase. If this is blocked by repressor is basically inhibitor, then gene will not work. So repressor is produced with the regulator gene which will block the operator gene, and if operator gene is blocked by this repressor, then genes will not function, they will be switched off. So this is regulating the other gene, so this is called as a regulator gene. Now repressor, as we know, this is a protein like we discussed earlier. This is a protein being synthesized by the regulator gene. This repressor can block this area and no transcription, no formation of protein will take place, no utilization of lactose will occur, and this can be only removed with the help of inducer. So repressor is a protein in nature which block the operator gene. And actually this repressor has two sides, one side for binding to the operator and other side for binding to the repressor. So two molecules of repressor are required to release the repression. Next is inducer. Like in case of Escherichia coli, we have seen a repressor is removed with the help of a substrate called as lactose. Now this lactose will remove this repressor. This lactose is acting as an inducer. Then RNA polymerase will further move over the operator and structural gene for transcription and translation, and three kind of enzymes will be produced. So this is called as inducer. Now this can be like in here it is lactose, then there can be other certain metabolite or enzyme can also be there which is acting as an inducer, or end product of a reaction can also be acting as an inducer. So this is about what is inducer. Now next is for the functioning of these regulatory genes, we need to have cyclic AMP. Now what is cyclic AMP? It is cyclic adenosine monophosphate which is being produced from ATP, that is adenosine triphosphate. This exert a positive control. Like we call operon system as Lac Operon because this is for the utilization of the lactose. So this is called as Lac Operon. Now it exert a positive control in Lac Operon because in its absence, RNA polymerase is unable to recognize promoter gene. So we can say like we have a DNA like we have seen here, it is promoter region, this is operator gene, right? So this is bound to the RNA polymerase. So the cyclic AMP will help in recognizing the RNA polymerase binding to the operator gene, promoter gene, sorry. So for its functioning, this also requires catabolite activator protein. This is catabolite activator protein which help in binding to the DNA. This is also called as DNA binding protein. So cyclic AMP is required and catabolite activator protein is required for the functioning of the promoter gene or binding of the RNA polymerase to the promoter gene or recognizing the RNA polymerase gene. So this is all about Lac Operon system of Escherichia coli, which was studied by Jacob and Monod. Now we'll overview summary we will study. Like first we have seen that RNA polymerase is recognized by the promoter gene. So this catalyzes or help in formation of RNA, helping in transcription. So genes will undergo transcription. Now those genes, now they will form with the help of ribosomal RNA or transfer RNA, they will form proteins or enzymes. Now here three enzymes they are being produced: lactose permease, beta-galactosidase and trans-acetylase. And for functioning of these enzymes, we need to have catabolite activator protein and cyclic AMP. Now beta-galactosidase will help in breaking of lactose into simpler component glucose and galactose, and lactose permease will help in entry of the lactose into the bacterial cell. Now beta-galactosidase or lactase, this is also called as an inducible enzyme. So inducible operon system generally occurs in catabolic pathways, mean when lactose is being broken up, when some metabolite is being broken up, this is called as catabolic pathway. So this is all about operon system. I hope you enjoyed my video. If you like my video, please like, share and subscribe. Thank you for watching.